Hello, everyone. Thank you for joining us today. We are your UniProt team for today. So I'm Emily. I'm a senior curator at UniProt. Daniel is a senior developer at UniProt as well, and Adura is joining us, is also a curator at UniProt. He's going to be answering questions and coordinating our question and answer session at the end. So I'd like to welcome you to Accessing UniProt for beginners and for students. Really brief introduction to UniProt. Hopefully you kind of have an idea of who we are to start with anyway, but our mission is to support biomedical research by providing a rich resource of always freely available and accessible, stable, comprehensive and accurately annotated protein sequence and functional information. We store primary sequences of proteins as well as of isoforms and also disease variants. We curate and store physiological protein function, and this includes subcellular location, pathway involvement, reactions, protein interactions, and any involvement in disease that we find. We also collate a lot of structural information, which includes topology and access to 3D structures. And we house data tools such as BLAST and multiple sequence alignment, a couple of which we'll be touching on later. So we have a few aims and general outcomes for today. We would like you to understand how to programmatically access UniProt data and how to pick the best approach for your workflow. We'd like you to become familiar with some of our tools that UniProt provides, with to help you programmatically access UniProt. And we're going to be touching on how to use Python to retrieve certain information. I will show you, and then Dan will show you how to work through these examples in Python.
Okay, thanks, Emily. So there's different ways to access UniProt data. The first one is FTP, and this is useful to grab everything we have at once. So it's like a big one-off download. On the right-hand side, you can see a particular directory that's available in FTP. So they're quite big files, and so when you download these, you'll need to do some post-processing, so parsing and sorting through things to grab what you need. And then we also have the REST API, which is what we'll talk about for most of today. And that is good for small to medium-sized downloads. And they are customizable, which will make more sense as we go through the presentation. But so there's many possible kind of annotations available for our proteins. And so you can customize which annotations you want to view, which fields you want to view. And these can be specified and encoded in the request that you make. And this is good for either a one-off or you have a pipeline, so you have a workflow you want to integrate into, you have a script. And then we also have website download, which is what Emily will talk about throughout these slides. And that's good for smaller one-off downloads. And again, these are customizable. And we have the newest edition, which is website file generation. That's where you have a really big result set. You have over 10 million protein entries, and you want to download all of them, and you want to specify certain fields. You can use this, so you submit the job, and then that will go to our backend, and they will build the file, and then they will produce a link to it, so you can then download it. And so this is an asynchronous process, but it enables you to have quite big downloads. So there's a lot of endpoints available within REST, and they mirror what's available within the website. So the main databases is UniProt KB, which is the knowledge base, and that's what we'll talk about today. And this is the most popular database. This has all the annotations, and there's two ways of creating annotations. The first one is manually curated or reviewed, and that's SwissProt. And then the other side is Tremble, which is an unreviewed, automatically annotated. So it's a computational means of predicting annotation. And then we have UniRef, which is pre-computed clusters of proteins at various levels of sequence similarity. We have UniRef 100, 1950. And then we have UniPark, which is a sequence archive. So for any given sequence that's been published before. A protein sequence has been published before. We have this in UniPark, and then we store the cross references to various other locations that also point at this protein, and we have a stable identifier for that. And then we have the proteomes, which is the set of proteins expressed by an organism. And then we have the supporting data, so taxonomy, keywords, citations, human diseases, cross-reference databases, subsidiary locations, and all these things are really just supporting the main databases on the left-hand side. And then we have some tools available here. ID mapping, which we'll have an example, and this is used to map from one set of IDs in some database to another database. So it would be with a database within, you can go from UniProt database IDs to some other non-UniProt database or vice versa. Go back and forth between the two of those things. We'll have an example to make that clear. And then we have sequence alignments for BLAST when you want to find a sequence within our database, or if you want to align your own sequences. And both of these are provided by the EBI service. So we're basically taking the job and then passing it to the EBI job dispatcher service, and they return the results to us, and we pass that on to the user. And there's also a peptide search. So if you want to have exact search, that's provided by our consortium members, PIR and Delaware. And then there's a few other miscellaneous databases. So we have Unirule and Arba, and this is utilized when we create the Tremble, so the unreviewed UniProt entries. And then we have Unisave, which is allows you to view the various entry versions. So as an entry changes over time, Unisave will record that change.
Okay, so for today, we've also prepped a live demo. So as I say, I'm going to walk through the website slides, and then Dan's going to show you how to do that programmatically. We're going to do it in Python. We've put together a Colab notebook so you can follow along. You will also have access to this after the webinar as well. So you can take this code, put it into your own pipeline, and you can edit it as you want. There's some explanation in our notebook as well that shows you exactly what you're doing. You can obviously access the API using a range of different languages as well. And at the end of the webinar, we will provide you with a link to the same examples that we're doing today, but also in the R language. So how to search UniProt. As you look on our homepage, you're going to find this big blue box and it says find your protein. You can use this as a free text search. So you can literally put a protein name in there, a gene name, whatever you want, or you can use the key value fields that are supported. So when you access programmatically, you don't actually need to learn the format of the fields. If you're unsure of what you're looking in, you can use the advanced search on the website, which is found in the first screenshot here. You click advanced and you get a little pop-up that is shown in screenshot 2. And the drop-down boxes allow you to pick the field of interest, and then you can either search using control vocabulary term or free text for what you're interested in. You click search and your results page will be shown, but also in the top bar, it will tell you exactly what search that you've just performed. So in this case, a free text for parking, and because you can use logical operators, taxonomy 9606, which is human. So your results table, if we are doing an advanced search for a gene name, in this case PRKN, which is colloquially named parkin, if you carry out this advanced search, you're going to get a results table much like this. Yours might look slightly different. You can customize this results table entirely as you want to show exactly what information you want to look at. I've left it reasonably simple for today's example. So here it shows you 676 results. You have a UniProt accession ID in the entry column, each UniProt name in the next column, and in the third column, the organism that it's from. And on the left, you have a set of filters that allow you to filter the results for different fields of interest. So you can look for reviewed or unreviewed, you can look for a popular organism, and there's various as you scroll down the page. So Dan's going to show you what this looks like in Python.
So this is a Colab notebook, and it's basically it's what we'll be using for the demo for today, and it's got all the work we're going to go through today. And the first thing is we need to do a bit of basic setup. So we're going to import some libraries. We use the sys library, which is we're going to use this when when we make a request, it doesn't work out, we'll just exit. And then we have JSON, which is what we're using to parse and uh, and print JSON objects. And we have request library, which is what we're going to use to actually fetch given a URL. And then we have pandas, which will provide us with data frames. And so this is the kind of root API URL we'll be using resting.org, and we'll use that throughout. We'll just be appending paths to this as we go through the different examples. And then this helper function is just wrapping around requests. So it'll take the URL of interest and then it'll get it. And then if it's okay, it will go to the response. If it's not okay, it will say something's gone wrong, which I hope we don't encounter that today. So I'll run that cell there. Mhm. Okay, and then down here we're gonna replicate uh, similar to what Emily has done, except we're just going to do the gene, the parking gene. We want. I'm not sure if the example that we had there. Yes, we're just going to do exactly what Emily had. So gene name parking, and uh, so this is the path that we're going to use, and this is appending the UniProt KB database, and then we're going to search, and here's our query, and when we request it, we can just pull the JSON out immediately from the response. But there's also other formats available. There's quite a few. So TSV, XML, and then text or flat file is also available. And if you visit this page, this help document, you'll see the list available. So we'll just pull out the JSON and then access the results object here. And then what we will do is we're going to try to reproduce the table that we saw in the website, and we're going to go through each of the results in a list comprehension and pass it through to a data frame. And we're going to pull up particular keys of interest, so primary accession, UniProt KB ID, and then we're going to combine two things here with the scientific name and the common name. So let's run that. And so here is the table that's been produced. And yeah, so the first column is primary accession, and we have the entry name, and then the combination gives you the scientific and the common name.
Okay, so now we're going to look how to retrieve a single protein entry and view all its information. So on the website, if you were to look at your search results that we just generated in screenshot 1, we just select the first row, which is parking in human. So you click the accession and it would open the entry page. So screenshot 2 shows you what the top of the entry page looks like. You have the basic information, you have the protein name, the gene name, whether it's a reviewed or unreviewed entry, and what organism you're in. And then below that, you'll have a function comment if we have function data, and below that, more information, etc., etc. On the left of the screen, you're going to have a set of titles. So you've got names and taxonomy, subcell location. They're all sections within the entry. When you get used to knowing where the data you're looking for is, you can click that particular subsection and snap straight to it. I've highlighted in the third screenshot here, catalytic activity for this particular entry, because we're going to show you programmatically how to filter this out. Because sometimes you don't want to see the whole entry. You're just looking for a particular piece of information. And instead of having to snap to a section of the page, you can filter that out using Python.
Okay, so let's do that now. Going back to the Colab. So we have this particular accession of interest, and we're getting the UniProt KB namespace, and we're just going to pull out the JSON here, and I'm going to show everything. It's a lot just to give you a sense of how much data is available. Yeah, there's a whole bunch of stuff here, a whole bunch of annotations and cross references. So for this example, we're going to focus. We're going to try to pull a particular part of the entry out, and we're going to just get the catalytic activity comments. So we'll do that by using the fields parameter here, and then we'll specify CC catalytic activity. And running this, you'll see it's much shorter. So you still get some of the fields. You always get the entry type and the accession, and then the comments is restricted enough to just the catalytic activity, which we saw. This corresponds to the screenshot that we saw that Emily just presented.
So as you scroll down an entry page, inevitably or at some point in your analytical protein life, you're going to be looking at some protein sequences. So we have a whole section on sequence and isoforms. We're still in PRKN, human entry here. So this is what that particular entry looks like. It has a canonical displayed sequence, which is always the first one that you get to. And it's identified by the accession -1 in this case. You can view the sequence, it tells you its length, its mass, and you can also download it in a variety of formats from the website. As you scroll down, you're going to see the different isoforms that we have. So we have 8 for this particular protein. This is showing the first 3, so isoform 2, 3, and 4. You can view these sequences, or you can just see them summarized here, which is what we've got on the right-hand side. And it tells you exactly how that sequence is different to the canonical, which is the very first one in the list. So you can see here, isoform 3 is missing an N-terminus, it's missing a C-terminus, and there's also quite a few residues in the middle of that protein that are different from the canonical as well. All these you can download. There's a download option to download just one sequence or all of them, and you can do various analysis on the website as well. Dan's going to show you how to pull these differences in these sequences out now.
Okay, so to do this, we're going to have to fetch a few different fields. First of all, we want to grab the sequence. So this is similar to the previous example where we wanted the catalytic activity. We've specified that field. We're going to do a similar thing, but we're going to specify different ones in this case. We're going to specify sequence. And then this FTVAR sequence, this is a feature which shows you all the sequence changes and the different isoforms through this particular thing called a variant sequence ID. And then we're going to have comments, so alternative products. So features are annotations that have been mapped to a particular part of the sequence, and then the comments are kind of like a global thing about the whole protein itself, like a global annotation. And so we use the CC alternative products to get all the possible isoforms, and then the FTVAR seq tells us the particular locations within the canonical sequence where things have changed. So let's go ahead and grab all those fields and we'll just print it out onto screen. And yeah, so now we have our comments here, and then below we'll have our features. There's a lot to see here, so let's do it step by step. First of all, we want to grab out, let's go ahead and get all of the different the features that will specify the different isoforms and the locations. So I've made this function here, get alternative sequences map, and that's going to iterate through all the features from the data we fetched, and we'll create a mapping from the feature ID to the feature itself. And we'll only do this for things that are alternative sequence type. And so when we've done that, we'll have this dictionary, which will go from this particular ID to the feature that we're interested in. And we're going to use this in conjunction with an isoforms object, which is made from the comments. And so this is basically all the isoforms and the names of them. So this would be the canonical, and these would be separate isoforms, non-canonical. And then we are utilizing the sequence IDs here. So we're basically be using these and looking up within the previous map that we just made. Yeah, so let's step through this. So we have this array of isoforms. We're going through each one, getting the sequence ID, and then going into the previous map and grabbing it out, and then using that to show the start and end location. And then if there's any difference there, it'll show you the original sequence and then the alternative sequence. So let's run that now. And this is looking like the website view. Uh, so for example, in isoform 2, between position 179 and 206, it's missing the amino acids that the canonical has. And then isoform 3, similar sort of thing from 179. And then 291, 297, there's been this substitution from these amino acids to this amino acid. And then lastly, we'll just grab the canonical sequence here by accessing the sequence key, and it just spits out the full sequence there along with different checksums.
So now I'm going to walk through a worked example of how to retrieve and filter disease variants for a particular area of a protein. So this is useful if you're looking for a particular, or you're interested in a particular domain or a particular site in a protein that you're working on. So again, we're in human PRKN, and we're interested in a particular domain called its ring type 1 domain, which is where it's active in the cell. So if you go to the family and domains section, as shown in screenshot 1 of the protein entry, you get a graphical view like this, where at the top you've got the amino acid string from N to C terminus, and you can zoom in and out on this string. Or you can filter down to the domains and regions within the protein string by using the type filter. Here we've selected zinc finger because we're only interested in zinc fingers. And I've just highlighted here that we're interested in the ring type 1 domain, which is between residues 238 and 293. So when we've written that down on our notepad, we're going to go to a different section of the protein entry called disease and variants. And this is what screenshot 2 shows. We have a similar graphic along the top where the amino acid string is on a measure, and you can zoom in, which is what I've done using the gray bar. So as you can see, we've got the zoom selected for the residue positions for the ring type 1 domain, and I filtered the table for natural variants, because otherwise it will also show you mutagenesis information, and that's not what we're doing in this example. So I filtered for natural variant, so that table will only show you natural variants that lie in that particular zoomed area or that region of interest, and we've got a number of them, as you can see here. This is all downloadable, so if you then want to download these variants and take them onto further analysis, you can do. Dan's going to show you how to do this using Python now.
All right, so we're going to kind of reproduce some of the steps that Emily did there, just using the REST API. And we're still in UniProt KB, and we're using the same accession. I'm going to change the field now to ft underscore z and underscore thing. So that's feature for zinc finger. And then fetching this, let's go ahead and do that. Just print everything to screen. So we've grabbed out the features. So each of these items will be a particular zinc finger feature. And you can see it has the start and end and then a description. And this is what's being used to populate the websites table we saw before. And so we have several of these available, and we can then also filter by using a list comprehension. So we'll iterate through each of the features, and if ring type 1 is in the feature description, we're going to pull it out and we'll save that. So let's run that code, and it produces a single feature. And what we're going to do now is we're going to use these 238 and 293, the positions, which Emily had mentioned in the previous one. We're going to use those to filter on the variants now. And we're just going to kind of traverse this object, going through the different keys to get the amino acid start and end values. And we'll call that query start and query end. And let's go ahead and print that out. So 238 to 293. And let's now pull out all the variants. So ft variant, and then gonna grab the features key there here, and let's just print it off the screen. and there'll be a whole bunch. Um, let's go to the top if we can. There's so many. Just start with one here. So here we have, similar to the zinc fingers, we had the start and end and a description, and we also have cross-references. So this particular variant feature has this dbSnip RSID, and we also have evidence available. So this would go to the PubMed ID, and then we have the particular change that's happened. So from a C to a Y. Now let's use the previous coordinates from the last step and filter these in the variants we just fetched. And we're going to do that by, again, traversing this object to get the particular start and end. And then we will just compare against the query start, query end, which we had derived in the previous cell. And then if it's happy, if it satisfies these conditions, then we'll go ahead and print that out. And so here what we have is we've printed the amino acid where the variant occurs at, and then the description. So this is in the Parkin 2 disease, and it has this dbSnp RSID. And this is producing the same rows that was observed in the slide that Emily just showed. And just to say, for Python, you can also do this sort of for loop. You can do it in list comprehension, which is a bit nicer. It's a bit quicker and more memory efficient. And this is the way you do that here. So it's iterating through all of these, and then if it matches its condition, then the final list will consist of everything that passes that.
So now we're going to talk about UniProt's ID mapping service. And as Dan mentioned earlier, this is a really useful service if you've got an analysis pipeline that gives you one set of identifiers, but your next step of the pipeline uses completely different identifiers. So essentially your two analysis steps are not talking to one another. You can convert UniProt IDs to IDs from other resources and vice versa. You can't go from another resource to another resource without passing through UniProt. So if you want to go from something like gene IDs to RefSeq nucleotides, you have to go from gene names to UniProt KBs and then UniProt KBs to RefSeq nucleotides, just as a note. So you can find our ID mapping service on the top toolbar of the website, and there's also links on the homepage. When you click that link, you're going to get a box similar to in screenshot 1 on the top left here. You can copy and paste your identifiers into the box, or you can upload them from a text file. You then select what you want to go from and what you want to go to. So we have drop-down boxes. We have a very long list of different identifiers that you can convert to and from. But in this particular example, we're going to go from UniProt KB accession to RefSeq nucleotide. So when you select them, then also name your ID mapping job. Just as a side point, if you're going to go from gene name, make sure you restrict taxonomy as well, or you're going to gene name search every single organism on the website. It takes time and you're going to get a lot of search results you don't want, so bear that in mind. But we're going to do ID, UniProt ID to RefSeq nucleotide today. And when you hit retrieve ID on those 4 identifiers, you get a results table similar to this on the right. You can download this and you can have a range of text formats and file formats on this. If you were to go the opposite way and go from RefSeq nucleotide to UniProt KBID, you would also have the option of pulling down all the UniProt information for those entries as well. So Dan's going to show you how to do this in Python.
Okay, so these cells, I've already run them just because I don't want the demo to break, just because we're depending upon another service. I just want to make sure we can view the cells. So these are already been run. So starting from the top here, we have the accessions that Emily had used, and you can also load whatever you want from a text file, for example, and just open it and then grab the accessions. And those will be used to pass to an encoded object, um, which specifies all the parameters of the job. So we do this post request, which is a little bit different to a get or post because we're going to post some data to it. And we're saying we want to go from UniProt KB, I'm going to go to ref-seq nucleotide, and then here's all of our accessions. And so that kind of defines all the parameters of the job, and we'll send it off to the API. And in response, we'll get a job ID, and that's this guy right here. And then when we check the status of the job, it will tell you it's actually running. And so if you pull this every minute or so, it will run, it will complete, and you will have the results. And this is going to be reflecting what we saw in the previous screenshot. So we're going from this UniProt KB accession to this ref-seq nucleotide ID. You can also, so this is JSON again, as most of these examples are, but you can also specify different file formats if you wanted to.
And now we're going to talk about BLAST. So to not go over BLAST in too much detail, all BLAST is a sequence similarity search. If you were to BLAST search your amino acid sequence of interest, you would search all of UniProt for a similar sequence and return similar sequence results. You can find BLAST all over UniProt, basically. We have a link in the top toolbar. We have a link at the top of every single protein entry in our tools dropdown, and that will search that canonical sequence that you're looking at. If you're in family and domains and in other sections of the website, you can BLAST just certain domain sequences, again, in the tools drop-down box. And then in the sequence and isoform section, you can BLAST any sequence that you find that's there. Again, there's a tool drop-down box and you select BLAST, and it will automatically fill the BLAST form with that sequence of interest. So when you when you click BLAST, this is what you're going to get. So this has automatically been filled. You can, in the very top box, you can add your accession ID, which will automatically fill that protein sequence into the BLAST search box. Select your target database. So this one is selected UniProt KB because that's what we're working in today, but we do have a range of different ones. And if at all possible, please restrict your taxonomy. If you don't, again, it will search all of UniProt. It will take a long time. You're going to get a lot of sequences back. So if you're looking at a sequence that's come out of an experiment, if you put human material in, it's probably a safe bet that's a human sequence come out the other side, right? So you can restrict that taxonomy. In this example, we've restricted it to mammals. Don't forget to name your job. And then when you run BLAST, it will open a tool results table like this with the name of your BLAST result and when you started running it. And it will tell you when it's completed. When you click complete, you're then going to get a results table that looks much like this. It's limited initially to 250 results. You can change that in the search settings if you feel like you need more. This overview shows you the entry accessions, entry name and organism, but again, you can customize these columns if you want different data back. On the left, we have different filtering options, so you can filter by similarity or review status of the entry or different organisms. And then if you click the blue bars on the right, where it's been sequence matched to the UniProt entries, you're going to get a zoomed in view of the sequence, and you can scroll along and see where your sequence is similar and conserved and where it is different. So in this case, the purple annotations, the purple amino acids are conserved, they're the same. And then where there's no purple and it's slightly gray, you see you've got an amino acid difference here. So Danny's going to run this same example. We're looking at myotubulin.
All right, so we're going to do a BLAST flow now, just like as Emily ran through on the website. Now, the first thing to do is, in order to BLAST, we need a sequence. So we will grab the sequence by specifying the FASTA format for that particular accession. And then, similar to the ID mapping job, we're going to encode this object, which will specify all the parameters of the job. of particular interest is um, the taxonomy IDs. So we're going to specify mammalia for this example. And then this is the database that we saw before. And then the particular type of blast is going to be protein. Um, and then this is where we say this is the sequence. So we'll just use this response text here. There's more documentation available at this page here. If you want to learn more about the other parameters of interest. And so when we run this, we will get a job ID just for the ID mapping job. We got a job ID, and then you use that job ID to poll the status endpoint, and so for this, it's still running. And this as well. This is the FASTA that we're using, and keep checking the status endpoint with the job ID, and eventually it's done, and then you get the results. And this is quite a lot of text in the response. This is a particular file format, where you can read more documentation about this here. We'll just go through it quickly. So there's kind of two main blocks. The first block here will give you a summary of all the things that it found and the score, the bit score. So this bit score is basically how good is the match, and the E value is the expected number of hits you'd expect if it's just randomly found. So it's 0. So that means that this is a very good hit. And on the left-hand side, we have found the accession that we actually searched with. So that's always good. And the SP here is SwissProt, and then the TR here is Tremble. So this enables you to quickly go through and see the reviewed status. So just to remind you, SwissProt means it's been reviewed by a curator, an actual human, and then Tremble has been derived through computational means. So its annotation has been derived through different rules. And so going to the second block now. So there's 250 of these results. You see the score is dropping as we go through. So this is our exact match because it's itself. So there's not a lot to be said here. This is a query that we provided, and this is the subject, which is what I found in the database that we specified. And whenever you have an amino acid in here, that means exactly the same. Let's find a slightly different example where it's of a lower quality, if I can. Right. So here, for example, we don't have exact matches here. We have a few. This is where there's a complete difference, and this is where we have a difference, but it's actually these two amino acids would be considered quite similar in a variety of ways, some physical chemical properties, or just through the scoring matrix that we specified before. So this was I think the Blossom 62. That is a matrix which determines the kind of penalties for a difference between two amino acids. So it's considered these to be fairly similar. And so all these things come together to produce the final score. And then also within these text results, you will have sometimes gaps. So this is saying that a gap had to be introduced here in order to create the alignment.
Okay, so these links I've put in the chat. If you want to scan the QR code now, we'll make sure these links are sent around as well after the webinar. We've got quite a lot of programmatic access documentation online on our website, so please make use of these. We are funded by the NIH and its various institutes thereof, as well as EMBL, Open Targets, and SERI. And we are a team all around the world at 3 different consortia sites. So me, Dan, and Adira are based at Emily BI in Cambridge, UK. But we have a significant number of teammates and colleagues in Switzerland at SIB and in America at PIR as well. So we have a lot of help and training. We do this throughout the year. We have a whole section on our homepage, and I put the link to our webinar series in the chat as well. But keep updated on our homepage and on our social media. Please follow us on LinkedIn and X. We have, if you click on tutorials and videos, we have a YouTube channel where we put on all our recorded webinars as well. We have loads of help pages on the website, so if you get stuck, please go and have a look at them. There's always a help tab on the right-hand side of your screen, and that will take you to the most relevant help page. And there's also a contact us button. If you get stuck, please send us an email. We do keep a very close eye on our email inbox, and we'll get back to you. We have a number of up-and-coming webinars scheduled. So on the 11th of December, we're looking at how data gets from the bench into a UniProt entry. In the new year, we have a structure webinar with our structural specialist, Connie. On the 22nd of January, we've got a tools webinar that looks at all the different tools that we run on the website. And then in February, I'm doing a UniProt for Proteomics Scientist webinar as well. Up and coming news, we've currently got new visualizations available on the website for GoCam. It's been integrated in the last couple of months. And we've got new documentation for our REST API, which is what we've been walking through today. And our next data release is expected at the start of 2026, which will be 2026-01. So I believe I do loads of training. I'm doing AJ's job here, but yeah, there's lots of UniProt stuff coming, and there's lots of other EBI training online. Please visit their training/webinars site, and AJ's going to pay me overtime for this. And all the webinars are recorded and on demand, so you can access them whenever you need to get some information or get some additional training around your time. Any questions?
Very good. Thank you very much. Thanks, Emily and Daniel, for the presentation, and Adura for helping with the questions. And thanks everyone for joining today. And just in case you missed any of the bit while Daniel was showing you doing things in Google Colab, this webinar has been recorded, and you will get the recording. You can watch it in your own time and try it by yourself. So, all right. So with that, we have now time for questions. There were a few questions. I mean, one question which was already answered by Adura in Q&A box. We have 2 open questions, which I will read out. So first one is, if I want to find out one specific function or attribute of a protein, how do I know what to put in the code? How specific must I write my code to only get one function or attribute?
Yes. Okay. So we have a whole bunch of different features that are available, and you're probably best looking at the documentation to see the variety that are available, because we encode all of these features with the fields. So like let's find one. FT sync finger, for example. You're best looking at the documentation to see what is available. And then you can use that information. You can know the field, and then you can request it in either doing this, or if you just request the whole object, you could then look through it as well. Your probably best idea is to go to the website first of all. If you are just kind of looking around to see what's available, you can more easily you can visually see this, and then once you have something in mind and you might want to go from that one entry to many entries, you can then you'll know what to look for within the response itself, and then you can filter down on that.
Great. Thank you. The next one is how to find intrinsically disordered regions using UniProt.
I think I'll have to take that. I had to check this actually. Look at Adura grinning. Yeah. So can I steal your screen, Dan? Because I've actually replace current share. So I've actually just looked this up. Oh, this share bar's in the way. There we go. Right. So in the website, you do an advanced search. Get rid of that. There we go. It's all right. I've got Zoom stuff in my way. So in the website, you do an FT region search. So FT is when you've mapped something to a particular amino acid on the protein. So you'd expect that because you're mapping a region. We map disordered regions to FT region line. They don't have their own FT disordered lines. So there's loads of filters on here. It's in family and domains. So you'd select region. I've typed disordered. And you search that, and it's just deleted my search, which is really nice of it. There we go. I've not put any other stringencies on here, so you can see we've got a lot of results. I've opened the first entry, and within family and domains, you've got disordered regions. So that's what they look like. They're usually automatically annotated, which means they're done using sequence similarity and rules and sequence conservation. So programmatically, you'd be looking for an FT_ region line with a comment description of disordered, I believe, Dan.
I'm sorry.
Programmatically, you'd be looking for an FT_ region, and the description within that would be disordered to pull everything out.
Yep.
Yeah. Yeah.
Great, thank you. And the next one is, how can one access the API key?
So there is not an API key, and it's all freely available and accessible. So all you need is the URL, the rest of uniprot.org. However, so it is free for access, but there's kind of a fair usage. So if you exceed a certain rate of access, then your IP will be blocked. That's just really to keep the service open for other people. So if we're hammered, then the service will go down and then nobody can access it. So we do have some rate limiting in place, but otherwise, as long as you're doing not too many requests per minute sort of thing or second, it's freely available. So no API key is needed. I hope that's what you meant by API key.
Okay. Thanks, Dan. I think the next one is also for you. It's I think it's a naive question that could you explain why one needs to use Google Colab or Colab Book?
Yeah, so you don't actually need to use it. It's just really for this demonstration. It's a nice way to collect all the commands we're going to run. You could run this in a variety of programming languages, scripting languages. All you really need to do is be able to make a request to an endpoint. So that's pretty much any language you can imagine. Python is nice because it's kind of a beginner friendly and it's quite succinct and powerful. And there's a large community that are supporting bioinformatics and Python usage. So it doesn't have to be Colab. It could have been something else. In fact, there's the R notebook, although it's using Colab. You can also do this you can make your own script on your local machine and just run it from the command line. Yeah, there's not really any restriction. You can go beyond Colab.
Great, thank you. Okay, so the next one is there's some feedback and then there's a question. So first of all, thank you so much for hosting this webinar. It is already answering many issues that I had before. I'm having problems integrating ensemble IDs with UniProt IDs. I just tried the online ID mapping tool with an example dataset and many were matched, which is great. However, some IDs were not found. Any recommendation on how to deal with the not found ones? Second question, is the online ID mapping tool the best solution or should we do it programmatically?
I'll take this one then. So if it hasn't been found, that means that the we collate a whole bunch of IDs through all of our cross references, and we try to maintain the latest versions that we have. If it's not available, there's the potential that the cross-reference has been removed, the link has been broken. So perhaps I'm just trying to think if UniProt could be of any use in that case. But I think we would already encode the information that's available within UniProt. So, well, that's a tough one. I'm not sure what to do if you can't find it.
I would say go to the source database and see if it's still in the source database. It's also worth noting that although we work very closely with collaborative resources, sometimes our production pipelines are not perfectly in tune. So Ensemble will do an update, we will get their data, and then that will be integrated into us. Same with Intact, same with Complex Portal, etc., etc. There will be a lag between the databases. So it could be that there's a lag between these cross-reference links being made, but it could also be that there's that the identifier you're using is older or out of date and has been deprecated. So I would say go to the source database to start with and check it's correct.
And there's a second part to that question. Is the online ID mapping tool the best solution, or should we do it programmatically? So I would say it is the best solution, either using the website or using the REST endpoint, because there's a lot of work going in to try to make these ID mappings as accurate as possible. But as Emily said, I guess if you encounter an issue where you can't map, go to the source database and maybe have a look around there.
We've got the same limits on the website form as well as programmatically. It's 100,000, isn't it, Dan? They're both the same. Yeah, so there's no difference in load, and you can upload a text file to both. Depends how you want to do it.
Great, thank you. If I have multiple proteins to target using mass spec, how could I use collab codes for pulling unique peptides already in the database?
That's a different API. That's the proteins API. I will put in the chat the link, and I will try and do that while I'm talking, to a programmatic access page. You want to use the proteins API. Within the proteins API, we have a proteomics pipeline, and you want to be using an endpoint within there, basically. So if I steal screen again, show this really quickly. Let's look a tiny bit of time. So on the home page, if you scroll right down to the bottom, we have a programmatic access tab. And within that, we talked today about the website REST API. So that fuels the website, but we also have a lot of our cross-reference data in our proteins REST API. It depends what information you want as to which API you're pulling from. We only gave you examples in one, so not to confuse anyone, as this was kind of a basics introduction. But for mass spec information for proteomics and peptides data, you're in the proteins REST API. And you want to be looking at the proteomics section, which if I just let that load 2 seconds, all the documentation for using this is up here. There's a paper here if you want to read it. There's a proteomics section, and depending what you want. So we've got proteomics PTM data here. We've got a generic proteomics pipeline is this one, which is non-PTM. And you can draw down from here using these fields. You can obviously access this without using this documentation. You could just access it pure programmatically if you want, but this is here for reference. And we've also got the Human Proteome Project proteomics data as well. And you can search within there and it will tell you whether them peptides are unique or not, whether we've got those peptides already in the database. And you can map them yourself as well using our peptide service, which is a peptide search up here. And that will tell you whether they're unique or not when you return the entries. And you could do that programmatically as well, but we haven't shown you an example of that today.
Yep, very good, thank you. The next one is, I think, a follow-up from the ID mapping question. So they're asking, I think Ensemble and UniProt, they always have a different version of data releases. So is it possible to set up like version-specific Ensemble or UniProt ID mapping?
So The ID mapping, I don't think will cover different versions. That would have to go to Unisave, which has the version history. But probably best in the situation if you could use the contact form, and you can show us what you're seeing, and we can better help you with that specific instance.